So today we are going to do transcription. And we're going to get a little sneak peek into Monday, January 25th's lesson, and that's going to be codons. We'll kind of put them ahead of the curve a bit. It's a good place. I'd rather be ahead than behind. All right, so let's do a quick review. Reagan, what does RNA stand for? Ribonucleic acid. Jasmine, what is the pentose sugar in RNA? Ribose. Wait, who's that young lady in the back? What's your name? In the Nike sweater? What's your name? I'm sorry, do you have a name? You do know your name. Okay, Bob. What are the 4 nitrogenous bases in RNA?
Uracil, adenine, and then cytosine.
That is correct.
Guanine or guanine?
Guanine. Now, Dan, when you're asking out your honey to go to prom, if there is a prom this year, you should say, if I could be a nitrogenous base, I'd be adenine, so I could be paired with you.
I will not be using that.
Guaranteed war. All right. Let's see, what else? What are the 3 types of RNA you need to know for this class? And we gave a description, like one of these serves as the blank. Which one of the 3 serves as the instructions to put a protein together, Jet?
What?
Wait, what? Turner? No, that's not the instructions. Risa?
mRNA.
That is correct. mRNA is the instructions. Which one provides the facility? Riley? rRNA. And which one is the only one left is the ingredients? Catherine?
The ingredients are
Stalling.
not
stalling.
that right here.
GQ, do you know what it is? Yes, tRNA. All right. So imagine this. You go to IKEA, you get yourself a new Verdal shelf or something. You're going to build it in your bedroom. You need a place to build it. That's the rRNA. That's the ribosome. You need the actual hardware, the pieces, the screws, the clamps, the wooden shelves, all that stuff. That's the tRNA. Those are the amino acids. And then you need the instructions. That's the mRNA. So if I had, you don't need to write this down. If I had a room like so, here's your bedroom, and you're a little kid, and you're going to learn how to spell your name. So we have, who am I going to pick on? Turner. So we got an R, and a T, and an R, and an N, and an E, and a U. We need all that information. So that's what the tRNA provides. The tRNA provides the ingredients. And then the mRNA will tell you how to put it together. That's you. That's how you write a name. Apparently you're missing the tRNA and the mRNA. Ah. Full of them. Never mind. Oh, I got dad jokes out the yin-yang. How did the hamburger introduce his girlfriend? Meet Patty. What is Bruce Lee's favorite drink? What's that? All right. You get it? Okay, transcription. According to dictionary.com, and this is not a biology word, this is an basic English language word. It is a noun, and it means a written or printed representation of something. So Reagan, I want you to write down what I'm about to say. Go Bucks, beat Green Bay. You don't need to really write it. But if she were to write it, what she would have in front of her is a transcription of what I just said. The verb is to transcribe. So in biology, transcription is using a DNA template to synthesize an mRNA complement. So this is really, I can really break it down into a 3-step process. In step 1, it's really not a step, it's more like a need. You require the template DNA strand that will be transcribed. So let's do a little drawing here. Mackenzie, if I have the gene sequence ATCGAGCT, what will the complement be for that?
It would be TAGCTCGA.
Correct. So you need your parent strand. Ooh, here's a good question. Notice that I have it laid down in a 2-dimensional depiction. What enzyme is keeping it flat on the ground and keeping it from twisting? Keeping it from getting all twisty. I did this one yesterday. Begins with a T.
Like the topoisomerase.
Topoisomerase. All right. Now, I'm going to highlight this half. This half is going to serve as the template strand. In biology, the template is what will be transcribed, which means this poor fellow over here is the non-template strand, and this is what will not be transcribed. So when it comes to transcription, everybody, you can essentially ignore the non-template strand. It has no purpose. You don't need it. You don't want it. Who cares? You only are going to transcribe one half. Yes, Ayla, is it?
Yeah. So this is the parent DNA right here, right?
That's right. Okay. So we've identified our template strand. Easy enough. Number 2. What enzyme is going to open up the DNA so we can actually access those nucleotides? Hartley? DNA helicase. DNA helicase will unzip the gene sequence. So let's basically just draw the gene that you guys did, but with plenty of space in between the two sides. A, T, C, G, A, G, C, T. All right. Now, one thing I forgot to do is label which one is 3 and 5 prime, so you can go and do that now. I keep forgetting that. There you go. All right. Now, pretty much third and final step. Here's your new enzyme for today. RNA polymerase. Will synthesize an mRNA sequence according to the DNA template. So I'm not going to bother drawing my non-template this time. CAGC, TCGA. Okay, so I want you guys to imagine this. This green enzyme I just drew, that's going to play the role of RNA polymerase. So watch this. So I want you guys to say aloud as a class, is this RNA polymerase scans, I'd like to think of it as scans the DNA bases. What will be the complement to those for RNA? You ready? So let's start with T. What would it be? You got it. So let's make our RNA. A, U, C, G, A, G, C, U. That is correct. This is what we call our mRNA transcript. This is what that sticky note was in my weird analogy about trying to get the cheat code. This is what's written on the sticky note, actually. It's the complement to the gene sequence that you wanted to transcribe. Now, let's go over a few of the basics when it comes to transcription. That's basically it for transcription for today. Because this involves DNA, where is this happening in the cell? The nucleus. It's like the bookstore, okay? So transcription occurs in the nucleus. That's where mRNA is made. mRNA, if you need to write that down, write it down. mRNA is made in the nucleus. I mean, that's transcription is the making of mRNA. It takes place in the nucleus. Now, mRNA is able to leave the nucleus, and I want you to think back to your semester review when you guys had to label like 19 or 20 different structures in the cell. What were those little openings that were all along the surface of the nucleus called? Jasmine? All right, so mRNA is able to exit the nucleus via nuclear pores. So just like you all have little pores on your face where sweat and grease and oil comes out, the nucleus has these little tiny pores that mRNA is able to escape through. If once the mRNA escapes the nucleus, it's now in the cytoplasm. mRNA enters the cytoplasm to join with, what is the site of protein synthesis? When I say blank, you say protein? Right. They're going to join with the ribosome. Remember that a ribosome is the site of protein synthesis. The mRNA is going to tell the ribosome how to make the protein. And this will lead to translation, which we're going to do next week. And translation will be a lesson that we will do, I think, next Tuesday and Wednesday. So that's pretty much everything for transcription for now. There are some parts that you will learn later about how the mRNA is refined and modified for life in the cytoplasm, but you don't need to know that today. We'll get to that later. Yes, Ayla?
Are more mutations happening in the transcription or translation?
Neither. It's happening in the DNA during semi-conservative replication. Now, remember something, guys. If this whole manual right here is DNA, what part, how much of it are you going to copy, not take, copy to form an mRNA? A little piece, a sentence, a paragraph maybe, a little piece. When Hartley sprains her ankle, she's running, she sprains her ankle, kind of overextended the tendons in her ankle, that's going to heal. She doesn't need the instructions on how to build an entire human being when you just have a pulled ankle. You just need the instructions on how to heal connective tissue. That's it. So when we are making mRNA, we don't need the entire blueprint of how to make a human. You just need maybe how to make some skin cells, or how to heal bone, or how to grow bone, or how to heal connective tissue, stuff like that. You don't need all of it. Okay, so that's it for transcription. And what I would like to do now is get into Monday's lesson. I'd rather be ahead than behind. We're going to do amino acids and codons. What are amino acids? Actually, I'm going to rephrase that. Are they monomers or are they polymers? They are monomers to what? Proteins, correct. Amino acids are monomers to proteins. Multiple amino acids form a peptide. Now, don't bust your brain too much. What type of bonds hold a peptide together? Jasmine? Peptide bonds. Held together by peptide bonds. Here's another question from Unit 1 of your semester review. How do you form peptide bonds? How do you get one amino acid and another amino acid to link together?
They like fold.
Not folding, that's forming a protein. But in actual, to get the two little beads, like the two amino acids to come together, what do you have to do to it? Andrew? Dehydration synthesis. How do you break those bonds? Hydrolysis. All right. Do you remember how many amino acids there are in nature? Good. There are 20. There are 20 amino acids in nature. There's basically two classes. There's essential amino acids and there's non-essential. Essential means that your body cannot make them So how are you supposed to get them in your body? Yes, you must eat them. Now, since there are 20 amino acids, 50% is 10. I know that more than 10 are essential. I think it's somewhere between 12 and 15. Let me see. How many amino acids are essential? Oh, I was wrong. There's 9. 9 are essential, so 11 are non-essential. So non-essential means that your body can make them. So you don't need to worry about getting them in your diet. If you don't get the essential amino acids in your diet, you're not going to be synthesizing proteins properly. And that could have some dire consequences because more than 50% of our mass is proteins. And proteins are, they are functional. They do jobs. They do things in your body. They're enzymes. They're structural as well. So if you don't have the right ingredients to make a protein, you're not going to have that protein. And that could actually jeopardize a lot of your cellular processes. Now, some of these amino acids have very odd names. And there's 20 amino acids. I'm not going to ask you to memorize all 20. I want you guys to memorize 2 names. 2 out of 20 is 10%. It's not too much to ask. But here's a few of the names I don't want you to bother memorizing, but I just want to show you how odd they are. You have some called isoleucine. Actually, it's I. You have leucine, histidine, lysine, valine. So there's 5 out of 20. The 2 that I do want you to memorize are methionine. It is not found in meth. Don't ask. And the other one is called tryptophan. Have you ever heard the word tryptophan thrown around at Thanksgiving before? Anyone ever brought it up? Well, I know they have in my family a long time ago, and I never knew what it was. But in case you want to throw some nerd knowledge out on next year, or this year actually, for Thanksgiving, let me tell you a little bit about tryptophan. Tryptophan is one of your amino acids, and it is plentiful in food like turkey and potatoes and stuffing, which is all synonymous with Thanksgiving. So it is plentiful in food. Especially Thanksgiving food. So it stands to reason a lot of food equals a lot of tryptophan. Now, what exactly does it do? When people mention tryptophan at Thanksgiving, they typically say, I'm getting the tryptophan coma right now. Here's what tryptophan does. Tryptophan triggers the secretion of a neurotransmitter. Yes, Annie? I don't know. Yes, it is. It is essential. Now, what is the neurotransmitter that makes you sleepy? Anybody know? Melatonin. Melatonin induces sleep. So if you have eaten a lot of food, you've eaten a lot of tryptophan, which means you're gonna secrete a lot of melatonin, which means you're gonna get a lot of sleep. Now, when you digest food, your body doesn't want to go to the gym. Your body doesn't want to do schoolwork. Your body wants to rest because a lot of your energy is being diverted to your digestive system. You literally lose energy from your other systems, your nervous system, your muscular system, your cardiovascular system in a way, kind of. It's all going towards your digestive system to break that down, which is why you get so sleepy and lethargic after a big meal like that. So it's helpful to just shut down the engines and let your body digest while you sleep. Now, speaking of melatonin, I want to tell you guys something else. So let's get back to amino acids, and here's what I would like us to do. We have about 6 minutes left. I want you guys to transcribe the following DNA sequence. Oh, my bad. I just had a brain fart. There. Okay, transcribe that. I want you to make the mRNA. I'll do it with you. So what you just did is transcription. What I would like you to do now is I want you to underline every group of 3. These are called codons. A codon is a 3-base sequence that codes for an amino acid. There are 64 different codon combinations, basically 3 cubed. So that means that there's amino acids that have multiple codons. Tryptophan only has 1 codon. I think it's GUU. I might be wrong on that. But you have other amino acids like leucine. They have 6 codons. It all codes for 1 amino acid. So I'm going to switch over to your PowerPoint. I'm going to show you what we use to actually translate the base, or not the base, it's the codons. So take a look at this table. It's a little overwhelming at first, but taking it stride. Where is my thing? All right. So look right here. I want you to look on the column on the far left. This is your first letter. I want you guys to look at the very first codon that you wrote. It's AUG. I want you to find the A row. So everything in this row right here is where you're going to start on the left. And then you work over to the top. You see it says UCAG. I want you to find U because our second letter is U. So you look down there where the A intersects the U. So we're now to those 4. And then you're going to look over here on the right to the third letter, which is G. And you're going to move and try to find where the 3 of those letters intersect. And that's going to be the amino acid methionine. Most proteins begin with the amino acid methionine. I have a little way to help you remember that. What month do we start school? August, A-U-G, methionine. That's how we start most proteins. So that's methionine. We don't have time for you to translate the whole thing, but we'll do that on Monday. But what I want you guys to see is look at tryptophan right there. Tryptophan only has 1 combination, U, G, G. But then look at proline. Proline has 4 right there. CCU, CCC, CCA, CCG. So some amino acids have multiple codons, while other amino acids only have 1, like tryptophan and methionine. That's translation. Now, another way you can read this is this chart. You start on the inside, you work your way out. So the first letter for methionine's codon is A. You start right here. You then go to U, right there. And then you then go to G, right there. You start on the inside, A, U, G. Forgot about that. And that's going to be how we translate. You're going to do an in-class activity with this tomorrow or Monday. All right, so